Thank you, Claire. It is truly an honor to be able to share my experience in developing and providing next-generation sequencing services for clinical research with you today. In fact, 2015 is a very special year in that it marks the 50th anniversary of Moore's Law and a decade of commercially available next-generation sequencing systems. In addition, President Obama's Precision Medicine Initiative helps us understand how next-generation sequencing, genomics, and big data promise to improve the way medicine is practiced. With the convergence of these various factors, it seems a perfect time to share with you how technology and some shifting paradigms in medicine can improve pathology and cancer research. So let me start with, again, some little additional housekeeping and disclose that the opinions I express here are mine and may not reflect the opinions of Illumina, other NGS-related providers, or even past or present employers. Furthermore, the use of products described or referenced here are within the context of laboratory-developed tests, or LDTs, and generally comprise research-only labeled components, systems, or products. Today, I will be highlighting just some of the benefits of integrating NGS into clinical oncology research and describe what this amazing technology can replace and what other technologies and applications it can enhance. I will then be introducing a case study from our most recent experience with integrating NGS and pathology services as we work to assist our clients and clinicians with advancing cancer research. And through our experience, I will further provide some specific considerations for implementing NGS in your laboratory and or clinical practice. In a sense, implementing NGS is a bit like playing a board game for the first time. We really need to understand the target players, the appropriate age ranges and number of players. We need to have all of the pieces and understand how they are used together. And finally, we need to understand and or establish the rules. I will highlight the key considerations of each of these areas, again, within the context of my CLIA experience. So before I jump into describing our recent efforts in integrating NGS into our pathology services, I'd like to provide you with a little background on our laboratory. This will hopefully give you some context with which to consider my comments and perspective, since each lab environment can be unique and a one-size-fits-all approach is not always best. And that's pretty critical. Otherwise, we can end up with solutions that are not well suited to our clients. So Molecular Pathology Laboratory Network was founded over 26 years ago and has served as a private, CAP-accredited, CLIA-licensed high-complexity reference laboratory specializing in pathology services and esoteric testing in the areas of oncology, infectious disease, and genetics. We are also one of only 8 U.S. labs accredited under ISO 15189. In 2005, we began providing clinical research services for IVD manufacturers and pharma clinical trials. 2 years later, we formally launched Genuity Clinical Research Services, a division of our MPLN business that's focused specifically on biomarker assay testing, drug discovery, and companion diagnostics-based services for pharma, biotechnology, contract research organizations, and medical device companies. Our integrated approach centers around molecular anatomic profiling, whereby we optimally assimilate services utilizing a variety of technologies and applications. For example, we offer IHC imaging, pathology, flow cytometry, fish cytogenetics, and PCR to fully support applications in oncology as well as other areas of clinical research. We see that there are at least 4 different sectors employing this technology. And it's important to keep in mind that there are many different types of laboratories out there. So I'd like to examine these 4 different sectors in a little more detail to, again, provide some perspective. On the left-hand side, we have private reference labs, such as ours, as well as hospital laboratories. These are generally considered clinical diagnostic laboratories, and they often offer FDA-approved or cleared laboratory tests in the form of IVDs or in vitro diagnostics, as well as laboratory-developed tests or LDTs, typically under the Clinical Laboratory Improvement Amendments, or CLIA 88. On the right-hand side, we have academic research labs and pharma-focused labs, such as contract research organizations. These laboratories may or may not be accredited by the College of American Pathologists, CAP accredited, and may or may not be CLIA certified. These types of laboratories may be defined more traditionally as clinical research laboratories. Currently, however, these traditional lines between sectors are blurring. For example, more and more academic research labs are obtaining CAP accreditation and CLIA certification in order to offer testing services to clinicians within their own institutions and even outside of their own institutions. In fact, several large academic medical centers have been instrumental in driving utilization of NGS-based testing for advancing pathology and cancer research and have contributed extensively to innovation around NGS library preparation, bioinformatics, and clinical annotation. So let's take a look at why next-generation sequencing is such a great fit for oncology research and why it makes sense to integrate NGS with pathology services. First of all, cancer is a genomic disease. Cancer was once viewed as a tissue or organ-specific disease, and most patients with a specific type or stage of cancer received the same treatment. However, it soon became clear that some treatments worked well for some patients and not well for others. Likewise, there were cases where patients received experimental therapies for cancers of different organ or tissue types from their diagnosis, and they had favorable responses to these therapies. These observational types of data, as well as the ever-increasing body of genomic data, has led to the understanding that cancer is associated with specific genes and pathways and less tied to a specific tissue or organ type. Several studies have provided evidence that complex genetic similarities exist across many cancers from many different organs or tissues. In fact, the 2014 paper by Daniel Heim et al. Describes the extensive analysis of genetic similarity from 14 major cancers using data from nearly 4,800 cases available through the Cancer Genome Atlas, or TCGA. They found that in about 43% of cases, tumors from a particular anatomic site are generally more similar to tumors from different organs and tissues than they are to tumors of the same origin. In other words, they are more trans-similar than self-similar. And this was the case for both carcinomas from different sites and also for neoplasms such as melanoma, acute myeloid leukemia, and glioblastoma. Further, because cancer is a genomic disease, we see many different types of variants associated with it, including SNVs, or single nucleotide variants, indels, insertions and deletions, amplifications, and fusions. And these cancer-associated variants are found across many different genes. A paper from the Genome Institute, Washington University in St. Louis, and Brown University examined the Cancer Genome Atlas, or TCGA, for point mutations and small indels from 3,281 tumors across 12 tumor types and found that 127 genes had significant somatic mutations. These types of studies and accumulating scientific evidence has led to a shifting paradigm in oncology research, one that places more emphasis on the genetic variants, genes, and pathways associated with cancer than on the origin of the tumor. Our improved understanding of cancer as a genomic disease points to why next-generation sequencing technology is such a great fit for cancer research. When we review these key features of cancer and examine the key strengths of NGS technology, the appeal of these methods becomes clear. For example, since cancer often involves variants across many genes, the ability of NGS to provide breadth and depth across the genome makes it an ideal technological solution. Because tumors can and because cancer can be associated with many different types of variants, the breadth and specificity provided by NGS is well suited to interrogating these different sequence characteristics, often without any specific a priori knowledge of their presence or location. And because tumors are also so heterogeneous, NGS provides the depth and sensitivity to recover variants present in minor cell populations, well below the 20% limit of detection of Sanger sequencing. For example, depending on the sample and library preparation methods employed, NGS can confidently assess variants at the 5% level and frequently even at the 1 to 2% levels. Finally, because cancer is a disease of pathways, the specificity and sensitivity of NGS allows for very targeted and highly sensitive interrogation across a number of different genes simultaneously. The technology is also a great fit for expression analysis and RNA-seq applications. So now that we've reviewed why NGS is ideal for oncology research, let's take a look at the potential tests and technologies that next-generation sequencing can replace. So this figure in a 2012 paper by Wall and Tonelato utilized the Association for Molecular Pathologists, or AMPS, solid tumor test directory to predict the network of test types and cancer types that could be replaced by whole genome analysis of next-generation sequencing data. The 45 nodes depicted here represent the most common tests and diseases in the directory. The 96 edges are drawn from test to disease if the test is used commonly for disease characterization. Using this mapping approach, the authors projected that of the 600 tests in use for analysis and clinical interpretation of solid tumors for more than 90 conditions, 85% could immediately be replaced with whole genome analysis for multiplex clinical annotation and medical impact assessment. While this projection of assay streamlining may not be quite where we really are today, it is clear that replacing single gene assays with next-generation sequencing-based tests confers distinct advantages to both laboratory workflow and clinical practice. For example, single gene assays may require more time, more staff, and resources, since each test typically only covers a single gene at best. Sometimes they don't even cover the entire gene often. In some cases, as with many PCR tests, only a few variants or even a single variant may be tested at a time. This can require multiple assay setups or even multiple methods. In some cases, practice guidelines and recommendations suggest serial or reflex testing, and that may require additional specimen and a longer turnaround time to results. If reflex or serial testing strategies are not optimized, this could result in missed opportunities for clinical actionability or available treatment options, and I'll discuss an example of this shortly. In addition, single gene assays are typically confined to more common variants and therefore represent a finite number of tests. In contrast, NGS offers such breadth that many genes can be tested simultaneously in a single setup and a single validated assay, which means more clinically relevant information may be obtained more quickly and without the need for utilizing as much specimen. NGS-based assays can again assess many variant types simultaneously and with better sensitivity than Sanger sequencing-based tests. The streamlined workflow with a single setup is particularly beneficial when limited precious sample is available, as is in the case of most solid tumor cancer research. These points help clarify the types of solid tumor assays that NGS technology can replace or streamline. But it's also important to look at the way NGS can be integrated with existing tests to enhance cancer research. Several well-respected institutions with pathology training programs have been critical proponents for integrating next-generation sequencing and genomics with other testing and methods to enhance cancer research. Their understanding that NGS methods provide an opportunity for molecular pathology laboratories to perform genomic testing that is far more comprehensive than single gene analyses, and that pathologists must acquire new knowledge and skills to fill the critical need of interpreting genomic results, led them to develop pathology training programs specific to genomic precision medicine. This diagram outlines the genomic medicine pathology training curriculum that was published and is freely accessible online from the group at Stanford. Here, their clinical implementation of a personalized genomic medical evaluation is a fully integrated approach. They state that the pathologist should be trained in emerging technologies to gain an understanding of the genomic test results and appropriately integrate that with more traditional testing, thus helping to accelerate clinical studies. Their program includes training residents on new tools, methods, and data from genomic science and integrating this genomic data with the results of clinical and traditional laboratory phenotyping or clinical phenotyping. The phenotyping may include bedside history, radiology or other imaging studies, physical exam, and non-NGS laboratory tests such as histology, cytology, immunophenotypic evaluation, and other laboratory and clinical methods. They believe that the discipline of pathology is well positioned to implement genome-based testing and to optimally interpret its results to drive clinical trials and new guidelines and standards. These types of training programs are helping to fill this critical need in personalized genomic medicine and helping us realize how an integrated approach with NGS and more traditional methods can help enhance cancer research. I think this may be an appropriate point to pause and see if there are any questions thus far.
Hi, Jamie. We do have 2 questions so far in the Q&A section.
Okay.
The first question, Dean, how do laboratories without access to pathologists or annotation specialists develop NGS assays with clinically annotated reports?
That's an excellent question, and this has certainly been a challenge, especially for smaller independent laboratories. Luckily, as I'll cover in a few moments, there are several annotation service providers that can be used, and these offer quite nice automated solutions for smaller independent labs.
Okay. The second question is, what do you think about the difference between DNA input requirements for various oncology kits?
That's an excellent question as well. So I think that's, again, I'll be covering some of that, and it really depends on, again, what the need is. I'll discuss, the need in terms of your clients and the end users, and that helps determine the content, which then in turn determines what variants and how many genes and what coverage is needed, which then in turn helps you select the appropriate solutions in terms of kits with different input requirements. So I'll be glad to cover that in more detail, and if I didn't answer your question sufficiently, maybe at the end I can address it more specifically.
All right, one more question came in.
Okay.
How important are microfluidic devices for NGS process?
Excellent question. Again, it depends on, your individual environment. So I think microfluidic devices for library prep can be very beneficial, and certainly in terms of when you can use a massively singleplex PCR approach to generate your library. The advantage of this is versus other amplicon-based panels is you may not have to worry as much about rebalancing your amplicons when you add or subtract content. So this can really streamline some of the development effort for tests that are working on laboratory developed tests, and it enables some more flexible content.
Great, thank you. That is all the questions for now.
Okay, great. So as you may recall, when I was discussing the advantages of next-generation sequencing methods over single gene testing, employing reflex or serial testing strategies, I mentioned I would discuss an example of some issues with this approach. So I'd now like to turn to a recent case study we encountered which illustrates some key points. This case study starts with an FFPE or formalin-fixed paraffin-embedded specimen that had been identified by a clinician as melanoma. As per the National Comprehensive Cancer Network, or NCCN guidelines, version 3.2015, the clinician ordered BRAF V600 variant testing of this FFPE tumor specimen. In this case, the clinician specifically ordered testing for BRAF V600 by real-time PCR. The assay was developed and validated as a laboratory-developed test, or LDT, to qualitatively detect V600E, V600D, V600K, V600R, as well as V600E complex variants. The assay provides a qualitative measurement of these somatic mutations in the BRAF oncogene and is utilized to predict EGFR inhibitor response in cases of colorectal cancer, hereditary non-polyposis colon cancer, papillary thyroid carcinoma, and metastatic melanoma. Published studies indicate that between 40 and 60% of melanomas have an activating mutation in the gene encoding BRAF. A phase 3 study by Genentech showed vemurafenib significantly improved overall survival in those with previously untreated BRAF V600 mutation-positive metastatic melanoma compared to chemotherapy. The specimen tested negative for all 5 BRAF V600 variants interrogated. Now, before I proceed with revealing more, let's place this case study on pause while I describe how we've implemented and integrated next-generation sequencing tests into our pathology services and how this integration ties into this case study. So this slide characterizes a highly actionable solid tumor NGS test that we have developed and validated as an LDT, or laboratory-developed test. The content of this panel is intended to specifically address the practices of our customers and clinicians, who tend to be community oncologists and pathologists. Obviously, as I've discussed, next-generation sequencing technology affords the opportunity to sequence across many more genes associated with solid tumors, but we needed to design content specific to the needs of our clients with their practices in mind. These practitioners, like many others, are not necessarily comfortable with interpreting genomic data and discussing it with their patients. They are not typically part of large medical research institutions and tend to refer primarily to NCCN guidelines in guiding their diagnostic and treatment decisions. For this reason, and because we were looking for the most highly clinically actionable targets to complement our existing menu and services, we selected content for a small, highly actionable 8-gene solid tumor panel from FFPE specimens. The genes in this panel are part of NCCN guidelines and would be familiar diagnostic targets for our clinicians. The panel has order and report selections for panels specific to cancer types. And while this may seem counter to the new paradigm in cancer research that I discussed earlier, keep in mind that published clinical guidelines and practices still primarily follow traditional cancer classification based on organ or tissue. Reducing the panel in this way through alternative ordering options also facilitates clinical decision-making by reducing the number of variants reported that may be of unknown significance in the tumor type submitted for testing. For this NGS solid tumor laboratory-developed test, we also wanted to ensure the turnaround time was no worse than our existing single-gene real-time PCR test. So, for example, since our BRAF V600 by real-time PCR test has a published turnaround time of 7 to 10 days, we designed our targeted solid tumor panel by NGS to also meet a 7 to 10-day turnaround time. And likewise, we validated the test for FFP specimens from slides and blocks. So let's dive a little deeper into the rationale for selecting this content given our laboratory setting. Again, we were looking for an entry-level NGS panel with a small number of highly actionable genes that clinicians would be familiar with based on the National Comprehensive Cancer Network, or NCCN guidelines, and also wanted that to follow their current ordering practices. We also took into account our existing capabilities and laboratory workflow in selecting the content and library preparation methods that would be the best fit for our environment. This table actually illustrates our selected content for this panel with regard to actionable mutations detected, clinical indication, and clinical relevance corresponding to therapy. These 8 genes have been identified by the NCCN, CAP, or College of American Pathologists, and American Society of Clinical Oncology, or ASCO, to be clinically actionable somatic mutations in solid tumors. The key mutations in each of these genes have also been identified through clinical trials. For example, BRAF V600 variants detected in melanoma FFPE specimens may indicate brimonidine treatment could be effective. By focusing on only this small number of highly referenced genes, we are able to save sequencing space for greater sequencing depth and to enable detection of variants at lower minor allele frequencies. In our development process for this 8-gene NGS LDT, we specified a priori the quality metrics that we were targeting and that the panel's performance would need to meet in order to proceed to validation. Some of these quality thresholds for variant calling have been published as guidelines by the New York State Department of Health. For example, New York State recommends a minimum score of Q20 for base quality and coverage with depth and uniformity to meet a minimum of 500 reads. Our assay development work exceeded these metrics with an average minimum base score quality of Q30 with a minimum of 2,100 map reads for each target, 50,000 map reads total for depth and uniformity of coverage. We also had a minimum of 30x coverage across all target areas. We further set our validation specification and performance verification to reflect a minimum limit of detection, or LOD, at 5% or greater mutation frequency in specimens. Even though empirical testing had demonstrated that we could reliably detect mutations present at only 2% in control samples. But it's better to be a little conservative when you don't know what kind of patient specimens you may encounter. So after selecting the clinical content and establishing our minimum performance standards, one of our next challenges came in selecting a clinical annotation provider. As the earlier question raised, clinical annotation in a reference lab setting can be a huge challenge since there may not be sufficient resources for manual curation for clinical annotation. Certainly, most smaller private reference labs will not have the pathology resident and postdoc resources available that the larger academic medical labs may have. Therefore, we look toward a commercial annotation provider, and I've mentioned that they're available in answering the question. And our annotation provider, we were looking for a more automated solution that could meet the needs of our specific workflow and lab environment. Some of the basic features we were looking for included the ability to support multiple NGS platforms, something that could be integrated with our laboratory workflow, the ability to customize yet still allow for rapid development, the ability to support multiple inputs and address multiple variant types, a solution that was compatible with interrogation of non-NGS data, a sound knowledge base with version control or change control capabilities, a technician-friendly interface, and of course we needed an annotation system that adhered to any compliance and regulatory guidelines we would be subject to. Finally, because we're still building our genomics program, we needed a system that could still deliver a branded report easily when we added new tests. So we looked for a solution that could generate new integrated reports through configuration but would not require significant coding changes. Of course, we can't overlook, that a major component of any genomic clinical annotation tool is the knowledge base. So because we are initially focusing on cancer-related somatic variants, we chose a commercial annotation solution that primarily utilizes MyCancer Genome Knowledge Base. The MyCancer Genome Knowledge Base allows us to link the disease, gene, and variant to approved therapies and also to available clinical trials. In addition, MyCancer Genome contains manually curated content for at least 21 cancers across 56 genes with more than 428 disease-gene variant relationships. MyCancer Genome, in addition to the MyCancer Genome knowledge base that our annotation solution provider utilizes, we also can integrate decision support provided by data derived from FDA, ASCO, and NCCN guidelines. So with the genomic content, performance specifications, bioinformatics, clinical annotation, and general laboratory workflow selected, we selected and developed for our solid tumor NGS laboratory test, we could go ahead and proceed with implementing our validation plan that was based primarily on the New York State Department of Health NGS guidelines for somatic genetic variant detection and CLSI MMO9A2 guidelines, among others. This slide summarizes the validation performance specifications of our highly actionable solid tumor panel LDT. Our validation studies were comprised of 49 different solid tumor FFPE patient specimens, 4 well-characterized FFPE standards, and 1 well-characterized molecular standard. Our results showed 100% accuracy across a reportable range of 23 exons in 8 genes with 75 confirmed variants. Precision studies revealed both inter-assay reproducibility and intra-assay reproducibility of 100%. Analytical sensitivity or limit of detection was validated at greater than or equal to 5% with analytical specificity of 100%. So now that I've described a little bit more about our solid tumor NGS panel and how we approach development and validation, let's turn back to the more general question of how to implement NGS to advance pathology and cancer research, which again may be unique in each different laboratory setting, if you remember back to the 4 lab sectors I introduced in the beginning. So I'd like to return to this concept of the board game and thinking about the players, the pieces, and the rules, as these can be critical questions. Excuse me. So, understanding the players is vital to successfully integrating NGS in pathology and cancer research because it is the players that are the ultimate users of the information from the lab. The lab's role is to generate robust, reliable, quality data and transform that into clinically useful information, as data in and of itself is not useful unless transformed into information. Because that information has to be useful, it can only be determined by the end user. If the customer or clinician cannot understand the information, the utility cannot be recognized, and the clinical decision-making may not be influenced by the laboratory report. Flashback to the slide of the Stanford Pathology Residence Training Program, where an integrated approach utilizes clinical phenotypic information, laboratory phenotypic information, and genotypic information, all synergistically guiding personalized genomic medical evaluation, and this helps underscore the importance of clinically useful reporting, which is ultimately determined by the end user. So when implementing the integration of NGS for cancer research in your own laboratory environment, it is critical to understand your customers. Are they internal, external, pathologists, oncologists, community practitioners, or research-oriented specialists? Are these groups, as all of these groups may have a different perspective on what makes the information useful. Ultimately, the type of information required by your customers or the players will define the application, the library preparation method, and the content. For example, some cancer research applications may require information on somatic variants, while other applications may be focused on constitutional or inherited variants. This in turn may guide choices in library preparation methods. For example, hybrid capture-based library preparation can work quite well for NGS detection of constitutional variants where plenty of high-quality genomic DNA is available. But in cases where FFPE specimens limit the quality and amount of available DNA for library prep, amplicon-based libraries may be the only good solution. And to refer back to the earlier question, this is one reason that microfluidic-based massively singleplex amplifications can be so useful. So likewise, what content is necessary for the application, and does it suit the need of the end user or clinician? For example, will interrogation and reporting of hotspots be sufficient, or will sequencing across entire exons or genes be required with the ability to discover de novo variants? In fact, deciding between a hotspot and exon sequencing approach should take into account several factors that will impact the development effort. This table illustrates some of the areas that will be influenced by choosing a hotspot panel or sequencing approach for somatic cancer research panels. The specimen type itself may affect the ability to offer a hotspot panel or sequencing panel. In general, most hotspot panels can work with a variety of different specimen types as long as there are no major PCR inhibitors present or they can be removed. Sequencing panels, on the other hand, may require higher quality DNA, and that may limit the types of specimens that can be tested. Generally, the nucleic acid input requirement for hotspot panels can be much lower than for sequencing panels. Even if we compare panels that both use amplification-based approaches, evenness of coverage is likely to be affected in a hotspot panel under limited DNA input situations. It's less likely, I meant to say, I'm sorry. It's less likely to be affected in hotspot panels. Of course, depth and coverage are typically higher in a hotspot panel approach because sequencing is focused on a very specific small region of the gene rather than dispersed across the gene or exons as in sequencing approaches. Because hotspot panels typically define the variants interrogated a priori, development, validation, annotation, and reporting can be less complex than for sequencing panels, and we expect the number of variants of unknown significance to generally be lower in hotspot approaches. Clinical actionability to variant of unknown significance ratios may generally be higher in a hotspot approach, since many variants addressed in guidelines and therapies are often of the single nucleotide type. However, there are certainly some applications tied to potential therapies where a sequencing approach is necessary. The bioinformatics requirements for a sequencing approach may also be more complex than for a hotspot panel. In fact, many commercially available cancer research hotspot panels available as RUOs, or research use only products, are available with a built-in bioinformatics pipeline. However, lest I give you the impression that a hotspot panel is always the best approach, it's important to understand that sequencing approaches are typically much more comprehensive and certainly are the best approaches for some applications. Allele dropout occurs, so hotspot approaches actually, while they minimize off-target sequence, they can be prone to allele dropout, which can result in false negatives. An allele dropout occurs when a variant is located in or near a primer binding site, thereby preventing effective primer hybridization and amplification and resulting in allele bias. While the risk of allele dropout can be reduced by using a tiled primer design strategy, assay specifications and performance with regard to allele bias should be carefully characterized with proper controls. Another benefit of sequencing-based approaches is that as new clinically relevant variants are discovered, these may be more seamlessly incorporated into the existing assay through validation and verification without the burden of a complete assay development. For laboratories offering services to support clinical trials, this could be an attractive feature. I'd also like to point to an excellent review written by Gray, Dunlop, and Elliott and recently published in Cancers. This review nicely outlines the nuances of solid tumor assay design for somatic mutation detection and may serve as a valuable guide in designing your own solid tumor assay. Now that we've examined how the players or clinicians affect the technical strategy used for developing a cancer research next-generation sequencing LDT, let's consider the pieces. The obvious pieces are the resources that will be needed to plug into the technical process of the next-generation sequencing workflow. These include support for the pre-analytical steps, the sample preparation and nucleic acid extraction, library preparation, sequencing analysis and bioinformatics, annotation, and the post-analytical steps. Despite this complex process and the ever-growing number of commercially available solutions, which might seem confusing, we have been able to, through the technology providers and their solutions, really this has resulted in giving us an easy button in offering solutions that link the steps in this process more seamlessly through the well-designed product lines and offerings. So, however, there are still some additional, not so obvious pieces that must be considered for implementing NGS. These include the instruments, people, IT infrastructure, and expertise, potential partners or vendors, and additional tests and services that the NGS offering will enhance or integrate with. In the current economic landscape with reimbursement and payment challenges, it is also critical to understand the market. As I mentioned earlier, the lines between clinical market segments are blurring, and understanding the competition and exploiting internal differentiators can be critical to success. For example, some private reference labs may grapple with the dilemma that the oncologist may be ordering the test, but the pathologist controls the specimen. In labs such as ours, where we also offer in-house pathology services, we have to consider strategies for effectively marketing to pathologists and help them understand that we are not the competition, but a trusted partner with the goal of integrating our laboratory services, both phenotypic and genotypic, with their clinical phenotypic assessment in order to help guide them in offering optimal precision medicine in a cost-effective manner. This goes back to the fully integrated approach of implementing a personalized genomic medical evaluation in pathology, which is the goal of several leading pathology training programs of today. So now that we have some understanding of the players and pieces in this game, let's take a look at the rules. Fortunately, compared to the early days of NGS in the clinic, there are now some excellent guidelines and other publications that help establish some key principles that should be used in developing, validating, and performing NGS as part of cancer research. The Clinical Laboratory Standards Institute MMO9A2 document is the approved guideline for nucleic acid sequencing methods in diagnostic laboratory medicine and may be particularly useful in that each chapter is designed to address specific issues across various applications. This document also serves as an international reference and attempts to avoid perspectives that may be specific only to local, state, or federal regulatory requirements. The New York State Department of Health Next Generation Sequencing NGS Guidelines for Somatic Genetic Variant Detection is also an excellent reference and imperative for labs looking to provide testing for patients in New York State. CAP has also published standards for implementing next-generation sequencing clinical tests, and these are particularly useful in understanding the test performance and workflow quality issues. Additionally, many excellent peer-reviewed publications are available that provide useful data and perspectives on development, validation, and performance of NGS-based cancer research assays. These guidelines and publications have essentially provided an easier button for labs looking to implement NGS in their setting. So, I hope I've provided you with a useful perspective on how the players, pieces, and rules fit together in these NGS approaches. And maybe before I return to continuing with our case study, I can see if there are any questions at this point in time.
Hi, Jamie. There are a few questions about the content that you just talked about. The
Great.
First being, what sort of QC metrics do you look at for your NGS test?
Yeah, great question. So now, I guess the thing to, I guess we did look at different QC metrics depending on, if we're in validation, and then we do, of course, monitor assay performance with standard QC metrics. There's two different sorts of issues there, and because there will be a standard set of metrics used in production as we offer the assay, and those are primarily based upon the standards and controls that we've included in the assay. So we look at general library performance. We look at we have low frequency variant controls that need to be above the threshold and that we expect. So there's really a variety of different controls, and keep in mind that at different stages in the process, it is important to use those metrics. So I'm sorry I stumbled a little bit, but keep in mind, some of the CAP recommendations are specific to a production setting where you're running the test over and over, and there certainly are quality metrics that you need to address before you, in development, before you move into validation or verification.
Great, thank you. The next question, How do you identify false positives or artifacts within your data? Can you recommend a specific variant calling algorithm that's well accepted and allows for assessing high-quality data?
Wow, great question. There's several options out there. Again, because NGS is such a complex process, you have to take into account the specimen, the nucleic acid extraction method, the library prep. It's really hard to find a good one-size-fits-all solution. So unfortunately, there's so many confounding variables. That's not a simple question to address without understanding, whether it's an amplicon-based or, hybrid capture-based approach and other features regarding the specimen type. I will say, in general, people look at NGS as being a highly sensitive method, but we can't forget that the real issue on quality and sensitivity comes from the specimen input. And so really you have to look at it within the context of the entire process or system.
Okay, thank you. We'll go ahead and continue the presentation.
Okay, great. So let's actually continue with reviewing some features of our targeted solid tumor NGS LDT. Again, in the content, the content is only focused on 8 highly actionable genes with order and report selections available across these different cancer types. But this is what we believe is most valuable and useful to our clinicians. So returning to our case study, you'll recall that we have an FFPE melanoma specimen that has tested negative for BRAF V600 mutations by real-time PCR. When we tested the specimen with our targeted solid tumor NGS LDT, we confirmed the sample did not have any BRAF V600 variants. However, the specimen did contain an NRAS Q61R variant that may have altered the treatment choice and qualified this patient for a clinical trial. So if we look, and I apologize, I should have left that part up longer. Essentially, with no BRAF V600 variant there, the patient was not a candidate for, that would have been counterindicated for the remover of therapy, and so based on NCCN guidelines. But actually, with the presence of the Q61R mutation, that could have really changed the treatment choice and qualified this patient for clinical trials. If we look at the report and how this sample would have been reported, we can see that, again, there was an NRAS Q61R variant, which was a missense substitution. It did have a pathogenic effect predicted, and that the therapeutic implications were not in the guidelines, but there was potential relevant clinical trials available. We did look at the mutation frequency present in this specimen, which was found at 24%. So this overview of our report gives you a sense of how we end up conferring this information to the clinician. We have obviously the results section with the genetic alterations and therapeutic implications at the top. We want to make sure that information is clearly visible. And then we have the interpretation section with our comments, and that is where we've stated the NRAS 182A to G variant was identified as well, and that that variant has been reported in 10 to 50% of NRAS mutated melanoma patients and confers resistance to BRAF inhibitors. But also is known to be associated with sensitivity to MEK inhibitors. We also have a section of the report that provides the drug response or predicted drug response, and as you can see, based on guidelines, there was no predicted drug response. However, in the next section, which I'll move to the next slide so you can see more clearly, we do report the various clinical trials that might be available. Sorry. So here we have potentially relevant targeted clinical trials, and we can actually sort this by region as well. So we have the capability to make sure that if a client is, for instance, in Tennessee, they will not be, they don't get a bunch of clinical trials listed that are in on the West Coast in California or in a region that's not attractive to them. So, and that again will depend on the number of potentially available and relevant clinical trials. So we do have a section where we can report other results, and this one happens to be a little more straightforward. We don't want to have a bunch of variants of unknown significance for our particular clinicians simply because of how they typically practice. We do have selected alteration details where we give more details on those variants and pathways if that would be of interest to the clinician. So, and this is just, I apologize, a larger view of that. So you can see the trial information for the prospective studies. Those might be available. And it's probably worth noting that there are clinicians who actually don't want to share that information with their patients. So for instance, a patient may be told that, there are some potentially relevant clinical trials available, but if they can't get into the trials, they then are, of course, not happy about that, and that can cause an issue for their physician in dealing with the patient regarding a trial they were not able to get into. So that may be something to consider as well, that just because that information is available, you may or may not want to share it with the clinicians, and your clinicians may or may not want to share that with the patients. We do have a section that lists the genes with no detected alterations. And this pretty much helps, this larger view helps you see what the report really looks like on the last page. So we, of course, will list all of the specific genomic regions as well for more sophisticated clients that want that information. And then, of course, we have the standard references, the CPT coding that's used, as well as any standard disclaimers. So hopefully I've given you some sense of how it really is fairly easy, especially now with all of the great solutions out there by commercial NGS providers, to really implement NGS in your own laboratory or clinical practice and integrate this with, both other molecular laboratory genotypic information as well as some of the clinical phenotypic information that may be out there. So, I really think, again, with understanding the players, the pieces, and the rules, it's really up to you to make the next move, and it's actually fairly easy now. We've had a decade of NGS. We've had certainly some sense of practice, and we've had life science and life technology companies that are really helping to make this a reality and simplify the practice for us. So I'd like to finally end, before I take any questions, with some key acknowledgements from my team here, and actually other teams I've worked with over the last 15 years. I've worked with some fantastic development scientists. So here at MPLN, we have our Associate Director, Alice Layton, who's done a fantastic job leading our development effort, along with Isabel Hubbard. And then other parts and members of the team include Jackie Seabrook, Dr. Annette Meredith, and Dennis Anderson. And if for some reason there's not time for all of your questions, I wanted to encourage you to feel free to tweet us at Genuity or send me an email at jplatt@mplnnet.com.
All right, thank you, Jamie, for an excellent presentation. For more information on Illumina's technology, you'll see in the web links, we'll have a Illumina NGS research page that you can click on.